One second, one second, one second. Is there a better one than this? If there isn't,
Thank you so much for letting me use them. I have this whole bag of stuff.
this kind of works, but it's dim.
The gift you're giving me, letting me be useful. Okay, here. There's
I just want,
this one, and then.
no, I don't need that. I just want to get a, for the point, laser pointer.
that- oh.
Yeah, might be, yeah. Where do I do press?
Green spot.
Yeah, that's good.
Okay, good.
Yeah, green is better.
Okay, so I will start here. So welcome everyone. It's my great honor to introduce this award and also see everyone here able to join this evening. This is ACS Philadelphia Section Award, and we have we have a chair. Matt is here, is the chair this year, and Graham is the past chair. I'm the chair of ELAC for this year and will be your chair for next year. I want to give a very brief introduction of this Philadelphia Section Award. This award is an esteemed honor recognizing members of ACS Philadelphia section who have made a significant scientific contribution to the field of chemistry. The award is given to individuals who work not only advance the discipline, but also enhance the public appreciation of professions. So I also appreciate Anne was leading the nomination, be a great colleague of Dan. So I will introduce Anne to give introduction to Dan.
Okay, well, I would like to welcome you all to Temple University Department of Chemistry, those of you who are visitors. So nice to see you all here. I am delighted to get to introduce my colleague Ron Levy for this well-deserved award, this Philadelphia Section Award. I am going to give the boring part of this introduction, so bear with me. Dr. Levy was an undergraduate at Reed College. He did his graduate work, excuse me, at Harvard University. And has grown his science alongside the growth of computational and theory, bioinformatics, and he's been a leader in that field for many years. He started his career at Rutgers, where he rose through the ranks to great renown. Temple was delighted to steal him from Rutgers in 2014, and so we've had him as a colleague for these last 10 years. And although he technically retired in December, he keeps telling me he's not really retired. He's just doing what he loves to do, which is research with his group. So many of whom are here tonight, and it's great for you to get to see him celebrate your work. Ron's research in theory and computation has been around the behavior of proteins in solution over many timescales. I think we're going to hear tonight about the richness of the work that he's done. He holds a Laura Carnell Professorship in Chemistry at Temple and has won awards including a Guggenheim Fellowship and a Fellowship of the American Academy of Arts and Sciences. He's also been a significant leader in the community of science, including leading NIH study sections and chairing professional science master's programs in our university. He's been a leader in so many ways. That was the boring part of the introduction. The exciting part of the introduction is that Charlie Brooks has composed a poem for the occasion. So I would like to invite Charles Brooks up to give the better part of the introduction, and then you can hear Ron.
Wow. It's an honor to be able to be here, to be part of Ron's award. We've been friends for a long time. And you know, I have this habit of writing. Answer your phone. I have this habit of writing poetry in a spontaneous manner. I wish I had heard the introduction before, because it's probably going to be sort of a boring poem, but here it is anyways. I wrote it sitting at the table upstairs. ACS sector, ACS specter, Halloween ghosts. For Ron we boast, toast, maybe roast his honor and recognition. Ron's worldview is igniting and science is paired to pots and lots of other proteins or drugs or stories we sweep under the rugs. But it's your night, Ron. We'll all have some fun. From stories of old, not so old, some retold with friends and colleagues. I'm glad to be here to share the delight on a near frightful night. Tomorrow, that's it, Ron.
Thank you, Charlie. Thank you, Charlie, and thank you, Ann, and thank you, Wei. So I do want to start off by, can we get the lights down a little bit in the? Not completely, but just a little bit.
Said this before, and then I forgot to. How's that?
Okay, that's, yep. And, no. Oh, here it is. So I want to start by thanking my department chair, Professor Ann Valentine, for nominating me for this award, and members of the Philadelphia ACS Award Committee for selecting me for the award. I was first informed that I had been selected by Wei Gao, who I met this evening, and that there would be a ceremony in October, and I was really pleased about that. And then 2 weeks later, I get this email saying, and by the way, you have to give a talk. Well, that's like, oh, that just ruined my fun. But I'm going to try to give a talk and fill it with some anecdotes and some science and try to make it not run too long. So that's my goal. The title of the talk is The Joy of Science. But I have to say, that's not my phrase. That's not the way I would describe science. So let me say a little bit about where that phrase comes from. Yeah. So there was an ACS symposium about 1.5 years ago in my honor that was organized actually by I don't think Charlie organized that. I think it was Carol Post and some other people. Anyway, there was a symposium in my honor, and there was a bunch of people who I've known for a long time and worked with many of them that gave talks. And one of the talks at that symposium was by my longtime collaborator who we've worked together, was Professor and is Professor Nanji Deng at Pace University. And this is his slide. And he started his talk with this slide, which shows my group in 2010. And there's Nanji, he's in the audience today. And there is me and other members of my group in 2010. And he titled the slide, Ron Levy, I use both pronunciations, the Joy of Science. And I was touched by that title. And maybe it implies I gave that sense to this group of people of the joy of science. But I wouldn't describe science that way. So let me say a little bit more about my feelings about science. And it comes from a recollection commentary in there was a special volume of Protein Science in 2016 that was put together by Charlie Brooks and Carol Post. And I wrote a little introduction to the volume, and I want to read both of these paragraphs that came from my recollection. My parents always wanted to hear me say that I find joy in my work, that it makes me happy. I was never able to give them the satisfaction of saying exactly that, To me, doing science is too intense an activity to be described in quite this way. Fulfilling, yes. Very stressful, definitely. I think they eventually came to appreciate my feelings and to understand that my career as a scientist has been deeply sustaining and rewarding to me. A friend of mine from high school days recently read that online. He's not a scientist. And he said, Ron, that's you. So that is me. That is the way I feel about science. It's something that is very, very important to me, very fulfilling to me. But joy, it's very stressful, very competitive. And that's the nature of science. Oh, I also wrote in this commentary in 2016, as I write this recollection, it was 2016, I'm sitting in my office on the 7th floor of a beautiful new glass and steel science education and research center with a wonderful view of downtown Philadelphia. I am excited by my recent move to Temple, which was in 2014. That was in that recollection. And this is the building we're sitting in right now. And it's a beautiful building. And I want to give a shout out to the person who made this possible, this building, and actually two people I want to acknowledge that played a significant role in building in recent years first-rate science and computational science at Temple, and one of them is in the audience right now. That's Hai-Lung Dai, who I believe designed this building and the building that's being built across the street for public health, who was the former provost at Temple, and Michael Klein, who is not here tonight, but is the former dean, and the two of them and their vision for first-rate science and computational science, I want to give some acknowledgement, important acknowledgement and tribute to. So the title of my talk is Exploring Protein Landscapes Using Computational Science. And there's a landscape, looks like a landscape that I think of conventionally. I'm going to be talking about other kinds of landscapes. And it's actually, it says, we moved there, my wife and I moved there from close to where Rutgers in New Brunswick is, which is a suburban area, to a rural area right near the border between New Jersey and Pennsylvania. That's the Delaware River. That's New Jersey. That's Pennsylvania. That's not actually the view from my living room window, but the view from my living room window is a beautiful landscape that looks quite a lot like that. So that's one kind of landscape. I'm going to be talking about a different kind of landscape tonight. But I also want to give a shout out to three people in the audience who were at the ACS symposium in my honor a year and a half ago. And there's Charlie Brooks, who people have been chatting with at the reception a little while ago from the University of Michigan, who's been, we haven't written a paper together, but we have to figure out a way to do that. But he has been a friend of mine and a colleague of mine for many, many years. I want to also mention that he is the 2020 ACS award winner in computational chemistry and pharmaceutical sciences. Something very close to that, something close to that. National award. Nanji Deng, we've collaborated for a long time. He was part of my group and has his independent group now at Pace University. He was also a speaker at this ACS meeting a year and a half ago, and Alan Haldane and I have been collaborating on things that excite me very, very much. And I'm going to be talking, he spoke at that meeting a year and a half ago, and I'm going to be talking about some of the work we've been doing together in a little shortly. I'll be talking about that. So, computational science. So the title of my talk is, what the heck is the title of my talk? Exploring protein landscapes using computational science. Well, computational science has been in the news lately, big time, including just a few weeks ago when the Nobel Prizes were awarded in both physics and chemistry to computational scientists. The Nobel Prize in Physics was awarded to John Hopfield. I would hope many of you know that name. A physicist of world renown who moved, I think he was at Bell Labs for a little while, but he moved back and forth for most of his career between Caltech and Princeton, where he traded departments from physics, molecular biology, back to physics, back to molecular biology, and renowned in this Nobel Prize award for his work on developing neural networks. Geoffrey Hinton, he's not really a physicist. I don't know if those two guys ever met. Maybe they did, maybe they didn't. But in any case, this is an award for, what is it? Using work on machine learning that uses artificial neural networks developed by John Hopfield, really mostly, and then further developments by the computer scientist Geoffrey Hinton, showing a completely new way for us to use computers. Think ChatGPT. But it's not only ChatGPT. It also has major implications in biological sciences. And in fact, the Nobel Prize in Chemistry is closely connected with the Nobel Prize in Physics. I know two of those. I know one guy reasonably well, David Baker from the University of Washington, who made a lot of use in the things he developed that awarded him the Nobel Prize for protein design from the developments of these people here. These are closely connected awards. I also know John Jumper, who is maybe a generation and a half younger, quite young. Maybe it's a little premature, maybe not. But in any case, these people were awarded the Nobel Prize for predicting and creating proteins, awarded to David Baker and Hasselbiss. I think he's a gamer. And John Jumper, DeepMind, that's the second award that week in artificial intelligence. And I know there are going to be at least a few people in the audience that are going to look at this and say, is that chemistry? Yes, it is chemistry. So, with that tribute to computational science, and I am a computational scientist, I was struck by the synergy between those two awards, the recognition of the importance of computational science and science today, and that's the wrong. And I want to say a little bit more about my computational science experiences. This is the first part of the talk. I am going to talk a little bit about science and my recent research projects, but by the time we get to that, maybe we want to stop. I don't know. So computational science, I have thought of my life in computational science as working on basic science problems, not paying too much attention to applied science, applications, or commercial possibilities, but I was fortunate enough to be quite friendly with other people that were paying attention to that. So this is a picture of four people on the scientific advisory board of this company, Schrödinger, that had its start in 1990. I was there in 1990. I remember its start. I was friendly with this guy here, that's Rich Friesner. He started the company. He had the vision for the company. I didn't have much faith in the company, but I was on the scientific advisory board for 25 years. And I remember the start of the company in Jersey City with 6 employees. And I was there on February 6th, 2020, when it had its initial public offering. This is a picture of Broadway and 43rd Street, where this is flashing on the street, on the NASDAQ building, when it had its IPO. And up until a few years before 2020, it was like, I don't think this is ever going public. I mean, it's interesting, and I'm participating in it. I'm on the advisory board, and I'll say a few more words about things that we've contributed to it. But it's this guy's vision that actually pushed it to become a public company, which it is. Let me mention a few more people on this slide. That's Bruce Byrne. Anybody in theoretical chemical physics knows the name Bruce Byrne. That's Bill Jorgensen. He'll be here in 3 weeks to give a talk in the chemistry department. I guess that's me. That's the scientific advisory board. I remember the 6 employees in 1990, and as of 2024, there are 850 employees in this public company. So I'm thinking basic science, but it has applied and commercial applications big time. And I didn't really think about it that way for most of my career. So let me, I don't expect you to read the titles of these papers. It's kind of a memory thing for me to say a few words about different periods of my life, my professional life, and the kinds of themes, scientific themes that I've worked on, at least some of them, that are embodied in these papers. So let me say a little bit about my beginnings. This provides an overview of the scientific themes that I pursued in my research. I was a graduate student in the group of Martin Karplus. I started as a graduate student in the group of Martin Karplus in the mid-1970s. But I actually did not get my PhD under his direction. That's another story. When I had a lot to drink, I could tell anybody who's interested. Most of my time was spent as a postdoc in his group. That was the late 1970s when the field of biomolecular, molecular dynamics simulations was first beginning to be developed. There were a group of us there in the Karplus group at the same time. Peter Roski, who gave a seminar here last spring, Andy McCammon, David Case, Bernie Brooks, and shortly after I left, Charlie Brooks arrived. So we didn't actually overlap. But these other people, anybody who knows anything about molecular dynamics simulations knows those names. And I have to say, I had no idea at the time when I was in that group of those people, those young people, that those are the same people that would be my friends and my competitors for the next 40 plus years. So that's quite something. I also note, as many of you, but maybe not all of you know, that Martin Karplus won the Nobel Prize in 2013 for his work on biomolecular theory and computation, not actually for molecular dynamic simulations, although some people, many people think that should have been, that was the real reason, but that isn't what's on the Nobel Award, but we won't get into that now. So let's see. Let me say a few words about this history of papers and themes. When I was in that group in the late 1970s, my first interests involved connecting molecular dynamic simulations with various kinds of experiments, particularly NMR experiments and X-ray experiments. And I and my people working with me did some of the first work in that area. This paper in 1982 with Attila Szabo, many people will know that name, was on protein dynamics and NMR relaxation and comparing the results of molecular dynamics simulations with experiment. It was published in Nature, one of my very few Nature papers. And it started me on a path in which I collaborated with experimentalists for almost my whole career. So there were a group of NMR spectroscopists that I was very close to at that period of time. We published a lot of papers together. And I've always felt that computation and experiments needed to be connected together from the very beginning. This paper, the second one here, I'm not going to go through all the papers, but the second one here may be the last paper I wrote with Martin Karplus. I was already an independent investigator by 1985 for 5 years. And it's molecular dynamics of myoglobin at 298 degrees, results from a 300 picosecond computer simulation. The first computer simulation of proteins done by Gellin, McCammon, and Karplus in 1976 was of pancreatic trypsin inhibitor in a vacuum for 10 picoseconds. 10 picoseconds, the whole run. This paper, we say in the abstract, this is 30 times longer and a much bigger protein in vacuum. A vacuum, that's not where proteins normally live. But that's what we did at the time, and that's what we could do, and now we run 300 nanoseconds, 1,000 times longer, and some of us with specialized machines run 300 microseconds, 1,000,000 times longer in the full environment with counterions and water and all kinds of things like that. So much more realistic simulations today than back then. So when I started my career, I started training graduate students how to use molecular dynamics simulations in the early 1980s. And my first PhD students got hired by Bristol Myers Squibb, by Merck, because Bristol Myers Squibb and Merck wanted people that had this kind of background at the very, very beginning of the field. And they worked there for a few years and then they were fired. No, that's not true. That's supposed to be a joke. They weren't really fired, but they were moved to other things because the power of the computer, in part, the power of the computers were just not there. And the promise of the technology was kind of premature for what the hardware was back in 1985. And it's taken 10, 20 or more years for the technology to catch up. And of course, many, many ingenious developments in terms of algorithms and things like that, in addition to the hardware speed and performance. All those things coming together creates not too many companies, but companies like Schrodinger with multi-billion-dollar capitalizations. So it was a little bit premature. Those first students were moved to other modeling projects at Squibb and Merck, but they're now every single pharmaceutical company has a modeling group that is doing something involving molecular dynamic simulations in one way or another. And this paper here, which is, I see Bruce Byrne is on this paper and Rich Friesner is somewhere on this paper. And this is a paper describing a program that was developed in my group called IMPACT, Integrated Modeling Program, Applied Chemical Theory. That program was about 200,000 lines of Fortran 90 code. I wrote about a third of that. So I don't know, 60, 70,000 lines of code. I wrote it. I haven't written code in 25 years, but I did write a third of that code. That was absorbed by Schrodinger. It was their prime MD engine for a while, several years, and they imported various sections of it into other programs that they developed. And that's both a plus for me and a minus for me, but it's more a plus than a minus. I mentioned that these first papers and the first simulations, there was no environment, there was no salvation. Well, that became another major interest of mine, major theme, the statistical mechanics and the statistical thermodynamics of solutions and interfaces mostly biological, but not all biological. This paper here, enthalpy, entropy, and cavity decomposition of alkane hydration-free energies, numerical results and implications for theories of hydrophobic solvation. No proteins in that paper, but it's one of my most cited papers. I'm kind of proud of it. And even from 1998, where we wrote a review article about solvation properties and using explicit solvent, to 2018, I've continued my interest in interfaces, solvation. We now use, I've had a long-term, he was my graduate student, but now he has been my teacher for a long time, Nobuyuki Matsubayashi. He was my graduate student, got his PhD with me at Rutgers, and now he's the chair of chemical engineering at the University of Osaka, going back to the country he came from originally. And he's taught me a lot about interfacial properties and density functional theory. And we still communicate, and published as recently as 2018 and 2019. I'm going to be talking about landscapes, free energy landscapes for proteins, unless I decide to stop. And what they mean for allostery, folding, and ligand binding. And I'll just say that in 2005, we published one of the earlier papers that on conformational equilibria and free energy profiles for allosteric transitions of this protein, ribose binding protein, in the Journal of Molecular Biology. I'll come back to that in this particular paper in about 5 or 10 minutes. We've done work on protein folding pathways and kinetics. Not very much work on protein folding. That's not been a main focus of mine. But this paper and this other paper highlights the fact that I've been interested in methods development for free energy simulations for a very long time, and using knowledge of statistical mechanics to develop algorithms. I am going to be talking a little bit about my most recent work, and it's with these people here. Joan Gizio, who's in the audience, and she got her PhD with me about a year ago or a little bit less. When was it? Where are you? Last year. Yeah. I'm going to be talking about this paper that we published in Nature Communications. Again, I don't have too many Nature type papers, but I'm really proud of this one. And I'm going to be talking about what I mean by machine learning and combining machine learning of sequences with structure-based modeling using molecular dynamics and conformations based on atomic models, sequence models and atomic models. What's the next slide? So now I'm going to get into it. I don't know how I may truncate this, but we'll see. So what do I mean by landscapes, conformational landscapes and fitness landscapes? And my focus in the last almost 10 years is putting these two kinds of perspectives on large biomolecules, putting them together, integrating them. So protein conformational free energy landscapes, those are something called potentials of mean force, and they're marginals. They're about figuring out what are the important coordinates of the problem and averaging over all the other, they're also important, but they're not the critical coordinates for some process, some reaction that you're interested in. You average over those, keeping the important coordinates, sometimes called the collective coordinates or the order parameters, fixed, and you map out what amounts to the free energy of the system as a function of these order parameters, which are, or potentials of mean force, and they're really a process of marginalization by holding certain things fixed and averaging over the other degrees of freedom of the system. Well, we can also think about that in sequence space. I'm going to say a little bit more about what that means. It's also a process of marginalization where we hold certain residue types at pairs of positions or fixed, and we average over what we see at all the other positions. So the idea of thinking about a fitness landscape in sequence space or a potential of mean force in structural space, they're both processes of marginalization. And I have been looking for ways to integrate those ideas in the last almost decade. So I'm going to talk about that in general, and then I'm going to talk about maybe our work on kinase family proteins. There's a picture, there's a landscape very similar to the landscape looking outside my living room window. There's another landscape. That's the back side of my house. That's my 1969 MG that I like to play with. And that's Carol Post, who came here a year ago to give a talk and ride in my MGB. So here's what I mean by potentials of mean force or free energy landscapes for proteins in conformational or structural space. And the simplest one you can think of is constructing the potential of mean force for the twisting or the torsions about the backbone dihedral angles phi and psi of a dipeptide, the simplest one, alanine dipeptide. And if we only think about just this isolated in gas phase or in vacuum, there's 66 degrees of freedom. But if we think about the fact they're greatly affected by the environment, which is the solvent or water, then we can think about the model should incorporate many layers of waters, or several anyway, and it's not really 66 degrees of freedom. It's 66 degrees of freedom, but plus maybe 1,000 waters or something like that. So it's 9,066 degrees of freedom. And the 2 degrees of freedom are the order parameters of the collective coordinates, and we're averaging over all the other 9,000 whatever degrees of freedom to construct this potential of mean force. And something like this was done while I was working at in the Karplus group, and they were running vacuum simulations of PTI for 10 picoseconds. But Peter Roski, working with Martin, was working on this kind of problem to map out the free energy landscape for a dipeptide, really taking into account what the environment really is. And here are some other ways to schematically think about the effects of marginalizing the problem, in which there are many, many degrees of freedom, and we're interested in some small subset of them. Tends to be 2, so you can plot it on a 3D graph or picture. We could be talking about protein allostery, about some chosen, hopefully wisely chosen degrees of freedom. We can talk about protein ligand binding in terms of the distance between the center of mass of the ligand and the receptor. We can talk about protein folding in terms of some generalized coordinates that involve energy and entropy. All of these things are marginalization processes in which we're averaging over thousands of degrees of freedom while we plot particular important degrees of freedom to construct the so-called free energy to move from place to place on that reduced coordinate space. You can do that also in sequence space. This is a multiple sequence alignment where we put together for a protein family. We strung out. Now, this is not coordinates. This is which residue type, one of 20 amino acids, is at each position in this one-dimensional representation of the protein sequence. And we can lay one after the other, one below the other, all of the sequence that represent multiple species of that particular protein. And we can hold certain positions fixed with a particular residue type and average over all the other residue types at all the other positions. If we do that, we get something called the bivariate marginal, which tells us how correlated changes are in residue types at two positions, maybe this one and this one. We can also think of higher order marginals, which is how more than two residue positions are varying at the same time when we average over all the other positions. And I showed you before a map of the potential amine force of alanine dipeptide. It kind of looked like this, with like there were hills and valleys. This is a map constructed by a guy named Arup Chakraborty at MIT. Maybe somebody in the audience knows this very famous scientist. But this is not a map of a potential amine force. This is actually a map of the likelihood of particular residue mutations as a function of averaging over all of the other mutations in the system, even though it looks like a potential amine force. And it's kind of machine learning because there are patterns. It's not kind of, it is machine learning. There are patterns in these multiple sequence alignments that we're trying to learn by a multiple variety of ways in terms of mathematical formulas. And one way to do this is with using something called Potts statistical energy models, which has this functional form that looks like something that came from condensed phase physics. If the possibilities at a particular position, instead of it being 20 amino acids or just two amino acid types, maybe polar or nonpolar, or maybe if we're thinking about condensed matter physics, it's spin up or spin down, then this is a model for spin systems in condensed matter physics. If it's just 2 degrees of freedom, if it's 20 degrees of freedom or more than 2 degrees of freedom, it's a generalization of the Ising model, and it's called a Potts statistical energy model. So the history of this model has its roots, the Ising model, in condensed matter physics. It was first applied to biopolymers by Anderson, and I don't know if Hopfield was on that paper or not. But anyway, this is a famous Nobel Prize, Phil Anderson, Nobel laureate, physicist. I'm not sure if he's still with us, but he was at Princeton for many, many years. Hopfield worked on neural networks, which is the kind of an alternative way to encode these kinds of patterns. And Hopfield won the Nobel Prize 3 weeks ago. There's been a recent, so this work goes back to 1983, using these kinds of Ising models for biopolymeric systems, starting in around 2008, 2009, there was enormous amount of attention refocused on using these kinds of models. Important players in this were Jose Anuchik, and working with Jose as the senior guy, junior guys, Farik Morkos and Martin Waite. David Baker, the guy who won his Nobel Prize 3 weeks ago, also was using, not very long ago, models that look very much like these Potts models. He's now migrated to other models that look more like neural networks. But at the heart of what Baker was doing was something very akin to what the Nobel Prize in Physics was awarded for. So the synergy and the integration between chemistry and physics this year is quite remarkable. Yeah. So I just want to, I may not complete this because I am going to stop, regardless of how far I've gotten in, 5 or 10 minutes the most. So one of the things that has been done in the mid-2000s with these Potts statistical energy models based on sequences was to predict the structure of proteins from sequence alone. Using something, constructing something called contact maps based on the strength of the Potts model lambda couplings between residue pairs. The stronger the coupling, the more likely those residues are in contact. And you could make something called a contact map just from sequences on one side of the diagonal and compare that for a particular protein with the contact map you could get from the protein data bank to see if the sequence picture of what was in contact actually agreed with the structure. So let me say a little bit more about that. I showed you in my list of publications a paper we wrote in 2005 based on molecular dynamic simulations, marginalizing the important coordinates to look at an allosteric transition of this protein, ribose binding protein, and we published this potential of mean force from molecular dynamics simulations. We constructed the free energy diagram in the third dimension or in these contours. It says how easy it is or how likely it is for this protein to be in a closed configuration, a metastable configuration, or an open configuration, all from MD simulations that were then projected onto these two dimensions that are the important order parameters for the problem. That was in 2005. In 2009 and to today, the Anuchik group and working with Terry Wah in those years, 2008, 2009, 2010, those years, were showing the power of these POTS models to predict protein contacts. And in a paper in PNAS in 2013, the title of which is Co-Evolutionary Signals Across Protein Lineages Help Capture Multiple Conformations, they studied some proteins that they knew had multiple stable states, and they predicted or they constructed from the multiple sequence alignment, sequences alone of ribose binding protein using the POTS model, which the contact map. So this is along the diagonal. On one side of the diagonal is the results from looking at the actual contacts from structures of the ribose binding protein taken from the protein data bank. On the left, on the lower side, or this side here, are the predictions of the POTS model constructed by Anuchik and his collaborators. And what they then referenced our paper that was 8 years earlier. I didn't know they were looking at our paper. I didn't know they were writing this paper. But they pointed out the consistency between the POTS contact predictions of where residues were in contact with what's seen in the protein data bank for the open state and the closed state. But they also pointed out they were seeing in the POTS model contact predictions a metastable state, which wasn't, reasonably enough, it wasn't in the PDB because it's not as stable as the open or closed state. But it was picked up in our MD free energy simulations, and it is in their contact map from their POTS model constructed from multiple sequence alignments. So this, in some sense, is a very important and perhaps the first direct connection between MD free energy simulations, which we have worked on for years since the 80s and 90s and beyond, and these machine learning sequence-based models, POTS models, that were pushed and developed by the Anuchik group and others in that time period. And the interesting thing is the POTS model is picking up certain contacts that you don't see in the PDB because it's metastable, not as stable as the open and closed states. And that gave us some impetus in that period of time to push further these integration between thinking about the problem in sequence space and in conformational space. I'm going to have to speed up here. So the problem soon after I got to Temple, I became interested in this problem of developing inhibitors of kinase family proteins. There's a class of inhibitor called type 2 inhibitors, which bind rather selectively. And the question is, how come they bind to certain kinases and not to other kinases? Two kinases which have very, very homologous sequences. And if you look at the receptor binding pocket, you can't see any difference really. Why does it bind to one and not to the other? That was a question I became interested in 2014. There were a variety of papers that were pointing out this issue. And this is a kinase in the so-called active or DFG-in conformation. DFG-in is a triple of residues, aspartic acid, phenylalanine, and glycine. It's right over there. And when the kinase is active, these type 2 inhibitors cannot bind. When it's inactive and the DFG triple of residues flips, then there's space for this type 2 inhibitor to bind. Even though the active site, oh yeah, so that's all I want to say about that. And the conjecture was that certain kinases can adopt that inactive state more easily than others. That was a conjecture by some people anyway. And this is just a map, again, order parameters. The order parameters are on the X coordinate. I just want to say on the right-hand side, in addition to this rather small DFG flip, there's a very large reorganization of a loop in kinases when any kinase goes from the active to the inactive state. At least I should also say that a very famous structural biologist wrote a comment in a science paper about kinases in Science in 2004. All active kinases are alike. They're in this part of this order parameter map of the activation loop displacement from some reference and the DFG displacement. But inactive kinases, there are many different kinds of inactive kinases. There's these here, there are these here, there are these here. And this group right here, in which the DFG is out and the activation loop is so-called folded, that's a big rearrangement. That's the class of, or the structure of kinases that these type 2 inhibitors can bind to. And there's a big rearrangement of the activation loop from the active state to the inactive state. And the thought is that maybe it's much harder for some kinases to do that than others, which turns out to be the case. We use Potts model threading to estimate. I won't go into all the details. I'm running through these because I really, it's almost 8 o'clock. It's almost 8 o'clock. I want to go to dinner. But the point is that when we did the Potts model analysis, we found that the preference or the conformational free energy balance based on not structure but on sequence for assuming the active state compared with the inactive state that the type 2 inhibitors combine to, it was much more stable, much more free energy favorable for serine threonine kinases to be in this active state than for tyrosine kinases. This is all, and there was analysis based on sequences and corroborated, and all of this work is really Joan Gizio's work that's sitting here. So thank you, Joan. And also the development of the Potts models, really Alan Haldane's. So thank you, Alan. But we were able to distinguish the ease of adopting the inactive state or functional state compared with the active state that distinguished STKs from TKs. But that's just the sequence-based perspective. And our interest was trying to see whether MD free energy simulations could give a consistent picture. And in two papers, one in eLife in 2022 and the other in Nature Communications that just came out, in two different ways, we showed that when you actually do the MD simulations, not to move reorganized the loop, activation loop by 20 angstroms. And I'm a big proponent of MD simulations, but I have serious doubts that whether that could be constructed accurately. So we played various kinds of tricks to get the same kind of result using ideas from junior level physical chemistry associated with thermodynamic cycles to estimate the free energy cost to go from active to inactive for particular sequences. What happens if you make a mutation? How does that change the free energy balance between active and inactive? And compare those results in structural space from molecular dynamics simulations with the predictions from the Potts model based on sequences. And we got remarkably good correspondence and consistency between those results. I'm almost finished. And in the last part of that work reported in Nature Communications, we connected our result with the evolutionary development of different binding modes of substrates. Of course, tyrosine kinases put phosphates on tyrosine residues on their protein partners, and serine threonine kinases do the same thing to their partners, but phosphorylating serines or threonines. And the substrates bind in fundamentally different ways for serine threonine kinases compared with tyrosine kinases. And we connected the results that we got from both the sequence-based modeling and the structure-based modeling with the different ways in which these kinases, I don't know what that is, but I'm gonna. Thank you. Okay, they're forcing me to, but I'm actually almost finished. So there was an explanation in terms of functionality of why serine threonine kinases differ from tyrosine kinases, the consistency between the machine learning Potts model viewpoint, the structure-based MD free energy simulations, very, very satisfying. That is, let me say, the end of the science portion of the talk. I think the anecdotes were more fun than the science portion. But anyway, I'm interested in protein landscapes. I hope you have a little bit of better idea of what I mean when I say landscapes and their processes of marginalization in both sequence space and potentials of mean force and conformational space. The machine learning of sequence patterns or Potts models. You can also use neural networks for the same thing. Captures not only the pairwise mutational correlations, but higher order correlations as well that distinguish tyrosine kinases from serine threonine kinases. And the Potts models and MD free energy simulations are consistent, very rewarding. TKs evolved to have flatter conformational landscapes, and it's related to their distinct substrate binding modes. We were able to do mutational scans with the Potts model to identify the effects of particular mutations on the free energy landscape and verify it with molecular dynamics simulations. That is the results reported in my paper published, our paper published just a few months ago. So starting off with MD simulations and NMR 40 years ago, this is what I'm interested in these days, and very excited about doing science, still very excited and very intensely committed to science, but I know an end will be coming. And I hope it's not me in a casket. I mean, eventually I will be in a casket, but I would like to do something else before then. So I've acknowledged my great gratitude and indebtedness to Alan Haldane for many, many, many scientific discussions and all of his insights and development of these machine learning Potts models. Joan Gizio, a very proud PhD in chemistry from Temple not long ago. And Abhishek Thakur was a postdoc in the group who worked on this. We have significant continued funding from NIAID to work on HIV and the evolution of drug resistance. Before I move, let me just finish off by saying Carol Post came to give a talk a year and a half ago. I'm so glad she came, because not only did she give a good talk, but she gave us some key ideas that were in this paper that we just published in Nature Communications about the relationship between what we were doing and these substrate binding modes. So thank you, Carol, for coming to give the talk and giving us some great ideas and working with us on them. I didn't talk about this other major theme in my research group, which has to do with drug resistance, the development of drug resistance, and the kinetics of the process as well. But I have some key collaborators, particularly Dmitry Limkus at the Salk Institute. These are the people in my group. This one is, she's not here tonight, but she is the spouse of that one there. She's there taking care of their child, I think, this evening. These are two recent graduates from the group who are now at the Salk working with Dmitry. And I hope I didn't take too long. Thank you.
And we do have a little bit time to.
Don't we want to go to dinner?
Questions.
Okay, no, go ahead. Okay. Sure, sure.
So, your focus on the kinase all about that.
Yes. Yeah, I mean, there's a lot going on with SH2 domains, and I know. Yes. Is that what you're working on?
So I was about to ask about the economy. I think it's actually I'm starting something. What kind of talk about also understand the functional sequence of
The answer is yes. We have a first paper. I mean, my collaborator is mostly focused on integrase, and so that's where our publications are in that area. He's a structural biologist, but we also have results for reverse transcriptase. And if you're working in that area, I'd be happy to chat with you about it. We do have results, but the publications are on integrase.
beautiful talk, but at the risk of exposing my ignorance, I got the impression that you are preparing your ND simulations based on calculating the landscape, and to justify the machine learning process. So the question is that how far can machine learning based on pattern recognition without I don't know if it's how much can it go?
Well, so I'm not going to completely answer your question, but it's a very apt current question. In using machine learning, mostly neural network based, the Nobel Prize in chemistry this year was given to a group of John Jumper that developed a program that you could access called AlphaFold. You give it a sequence, it gives you a folded structure. It's not free energies, it's not thermodynamics, but figuring out with neural networks, where's the physics, is extremely difficult, maybe impossible, okay? We use these POTS models as an alternative to neural networks because the parameters in the model have much more physical meaning. So the issue of how do we ascribe physical meaning to machine learning models in part has to do with what models are you using. And I wouldn't say we use the MD to justify the machine learning POTS model results. I would say that you can, with a little bit of cleverness, you can think about what is the corresponding kind of properties you could calculate with the machine learning models that make sense to compare with the free energy simulations, if you will. And there we see this correspondence that I find very, very satisfying. That's all I want to say now. Certainly.
So I'm not a protein. I think we picture polysaccharide also is a big piece. How do you see those models for the polymer, like polysaccharide or any other polymer, if we gave the structure, if the branching functional, how far that?
Well, I'm going to answer part of it, and then Charlie's going to answer the rest of the question, okay? Well, you have to repeat it for him. I'm going to say that a lot of work has been done modeling proteins embedded in membranes and transport proteins and the role of the membranes in atomic detail. So the role of lipids and how they affect the formation of the membrane and what's embedded in the lipids in the membrane. There's I'm not in that field, but there's a robust field already there. In terms of the role of polysaccharides, which Charlie, what do you have to say?
Polysaccharides, polysaccharides that catch post-translational modification.
Yeah,
Just in general?
just in general, even very high polysaccharide, not just the shorter one that really.
I don't know anything about that either. I mean, I can say that the polysaccharides that come as post-translational modifications of proteins are really just a way of
Yeah, thank you. Anyone have a more question? Yeah.
Can I still ask a second question? It's so inspiring. So, it's probably polymer, like protein chemist, actually ever since Jim Wells at UCSF have identified the proposed concept of allosteric sites. All these particles are actually trying to identify allosteric sites, and there is no perfect solution yet. As you mentioned also later in your talk, you practice pulse modeling to identify some capillary sites and some new sites being never observed in X-ray in crystal structures. Do you think, has anybody ever used pulse model like machine learning type to predict these allosteric sites on the protein?
I think the answer is probably no. That one example I was showing where there is a correspondence between what's seen in a contact map coming from the POTS model and what we did many years earlier in constructing a MD free energy diagram was kind of remarkable to me. And it kind of set us off thinking about the close possible connections between MD free energy surfaces and machine learning results predicting structures like AlphaFold. But I doubt that anybody is doing what you're asking right now, but it's very primed to do with the right kind of biology background and the ability to kind of work with AlphaFold. I think there's a lot that could be done.
I think with the time limit, I will stop everyone here. In half hour, we will have a dinner at after. I welcome everyone go there, pay by yourself. I think they also accept the walk-in order. We have some room booked for the people already registered. But I think a very healthy discussion, question. Let's send Ron again.
Decibels for the people that are not kind of. Yeah, I was thinking about it. Yeah.
Grab the. Here?
Oh, sure.